I hope everybody can hear it. Hi, everyone. I'm Jay. I'm from Liz Lab, and I also worked sometimes at the MSET. So today we're going to talk about the 3D correlation strategies for FIB-milling, and also we're going to combine with Cryo-ET. So just to start with, we know Cryo-ET is a very powerful technique, but is also very likely you're looking at a very crowded environment and trying to target where you are trying to look at. And you can just see that a very sneak peek of the nuclear peripherals of the HeLa cells, and then it has like over 200 proteins. And also even for very simple and elegant E. coli system, you have a very crowded environment. So the question comes is where and what you are looking for. So targeting becomes very important. So we're going to talk about targeting in a very detailed way, hopefully, and then we're going to talk about 2D and 3D correlation. We're going to talk about factors associated with the precise correlation, and then also along the way, we're going to discuss some recent advances in the field. So the 2D correlation. So when you're talking about correlation, generally you will involve the process called the image correlation. So you will start with registration, transformation, and correlation. So what happened is that you need to pick a set of known corresponding points from both your source image and your target image, and to use other registration points. And from the registration points, you can calculate a transformation function. And using that function, you can apply the transformations to your source image to get a transformed image back projected into your targeted map. In this case, is a TEM. And because you correlated two images, your FLIM and your TEM, so you will be able to identify your fluorescence signal. And in practice, there are several things you need to consider, and there are actually more. So I'm just going to list a few things that we're going to discuss today. So the first is precision in registration, like how precise your registration needs to be. And then we're going to talk about transformation function, because there are linear and non-linear. So which way actually works better under cryo. And then we're also going to talk about the FLIM, because there are FLIM can happen before freezing and happen after freezing, and you correlate with your TEM. So is there any difference? And then we're also going to talk about correlation on the fly versus post, because when you are doing data collection, you want to do it on the fly. And then when you already have your data and also have your FLIM images, you can do some post-acquisition correlation to know what you're missing. So the registration, as I mentioned, like how precise the registration needs to be. So when we come to registration, one of the most popular targets is using fiducial beads, and generally would be the size of 100 to 200 nanometers or even smaller. And then, so as you can see through this linear intensity profile, that the background peaks actually bleeds into your ROI peaks. So if you just have a raw image, you may have some influence from your background, and then you need to do some pre-processing. And in this case, you need to denoise your background, and then also you can do some maximum intensity projections on the top of the denoising and background removal. And then you can do some other processings like extended depth of the field to actually clean up the peaks. The cleaner the peaks is, the more precise you can actually pinpoint where your coordinate is. And also this cleaning is kind of important, not just for the registration. You can also see that with a cleaning background, you can identify your ROI easier. So this is a case that we applied on our CryoClean wide field system, the Cryo Thunder system, that we wanted to identify the edge of the cells where the RSV is actually budding, which is a green signal. So compared to the raw images actually on the top and on the left, which is the C and D, you can see that after cleaning, the image is much sharper and I can pinpoint where I want to look at. And then the registrations. So there's no perfect microscope. There's always some like chromatic aberrations, and there are always some misalignment that exist in your microscope. And then so when you're actually using fiducial beads, one thing you will notice that your fiducial beads is super bright. And then that can actually blur your ROI and makes the pre-processing very challenging. And then also another thing is that the fiducial concentration needs to be optimized. So we and also other group found out that if you're just looking at quantity quantifoil grids and looking at the 2D correlation between film and TEM, you can actually use a whole centrals. It's more tolerant to the coordinates misplacement, which is where you want to place the center of your fiducial. And also it are more tolerant to the chromatic aberration from the film signals. And also holes are everywhere, so you can pick as many as you want. And we actually did this quantification to see what kind of the chromatic aberrations between the bright field and then the fluorescent channels where we're interested in the pixels misplacement, and it's actually very minimum. And then the question comes to how many registration points you needed, because there are like a lot of holes in a square. So do you need to pick a lot, and how you pick them? So we found out that if you pick a set of 5 hole centrals at the registration points, it actually can achieve a pretty decent transformation. And then this is actually using the leave one out method. So briefly, leave one out means that you will pick a set of registration points, but leave one of them out as your ROI. So you will use that because you already know that is going to be your ROI and know where the correct location would be. So you can actually measure the deviations between your real ROI and your predicted ROI. And then we also wanted to know that right now we have like a set of the points and then where we should position them. So the distribution turned out to be actually more important than number of the points. So if you have a really restricted areas and then to place those fiducial beads, you actually will get much worse correlation despite how many fiducials you have. So you want it to be close to your region of interest, as also you want it to be spread out. And then we need to talk about transformation. So transformation is actually it's a function. So what is happening is that it relies on a set of the basic transformation functions where you can use it to transform your image projected onto your target image. And during this process, your image will be transformed, which is going to deformed in different ways, and depends on the preservations of the linearities of the lines, which is the points coordinates in the image. You can have a linear manipulation and versus non-linear manipulations. And then strictly speaking, translation is not a fine transformation in the math, but it's actually very likely to be grouped in the fine transformation because of the linearity preservations in this process. So you will have like rotation, scaling, and shear, and the reflection, and all of them combined will give you the right transformation function. And also you can see that for the projection, the projective transformation, your linearity is preserved, but 2 lines actually are changing directions. And for the nonlinear transformation, generally it happens is that your lines, once it is straight, it's not going to be straight anymore. It's going to give you some deformation, which is having this curved line. And then people have found out that because of the vitrification, it's actually limit the physical deformation of the sample. So in most of the cases, a fine transformation works for the cryo-EM, especially under 2D. So we also wanted to see if it's actually the case. So one thing about the nonlinear transformation is that you do need a lot of registration points because it accounts for different coefficient factors in the function. So we found out that because you are using holes, so you can pick a lot of holes, so registration points are never going to be a problem. So we wanted to know whether or not you can gain any benefits by applying the non-rigid transformation. And here we use TPS, which is a very common nonlinear transformation applied in the field. And then so what we are doing is that we have the same set of the ROIs and we're going to use, and then we image them under RT film, and we also image them under the cryo-film, which is we freeze them, and then we load it onto the TEM. So we're going to correlate. So we're going to see that before the freezing, after the freezing, if the RT film to the cryo-TEM and the RT film to the RT-TEM and cryo-film to the cryo-TEM and RT-film to the cryo-TEM, is there any benefits to apply the TPS? And what we found out that there's no significant difference in the prediction errors. And then we also want to see how many points, as I mentioned, that the points actually play a role as in this TPS. So I was wondering if I pick like 5 registration points versus 10 registration points, will the 10 registration points actually give you better results? And what we found out that there's no significant difference in this process as well. And then actually other group also reported that if you apply non-rigid transformation when it's not required, it actually gives you worse registration errors. So you can see that the bottom, which is those green lines, are actually the ground truths because they're also using the live one out validation. And if you apply the nonlinear transformation, in this case, they also use the TPS, it's actually at every point, it gives you the errors. So if you don't have it, don't use it. And then the second part is that the ice thickness, because under cryo, you have the ice. And then people always wonder, will ice actually also introduce some deformation? Not particularly around your sample, but probably obscure your signals or something. And there's a lot of discussion as to how fluorescent signals behaved under cryo. And we are not going to discuss it right now. So we are looking at ice thickness as to the precision relocation. And then what we found out that when we're talking about wide field, which is a lateral resolution of the cryofilm is around like 400 to 500 nanometers, there's actually no significant difference when it comes to a thicker sample versus thinner samples. And then how we measure the thickness of the ice is actually we use a very popular way, which is called a apparent mean free path, which is measure the inelastic scatterings before and after the energy filters. And then that is actually corresponds to the ice thickness. As your electron goes through the thicker ice, the more inelastic scatterings is happening. And then so just summed up during this general 2D cryofilm to the TEM correlations. So you will actually end up doing the image correlation twice. You will do it first on the square level because you just need to identify which squares you are looking at. But you also need to know exactly where you are looking at inside of the square. So we will do this one more time and apply it and then collect the two series. And then we also wanted to shamelessly plug our software, which is a correlator. So what I mentioned this is because we wanted to actually know the on-the-fly correlation. It becomes very important for us to actually collect where we want to collect, considering how many hours you need to spend on the data processing and data collection. So in this correlators, what is happening is that we will have the image, we will have the coordinates, and those coordinates will be directly linked to your stage position. So after you do all of those transformations and you get correlated images, and you can just pinpoint where you are looking at, drive the stage there and collect the data. And all of those information are present in the exported files from the correlator. So how does it work is that we work with the cryo-EM because it's very powerful, very popular, everybody loves it. And also it is it has a one advantage is that all of the stage coordinates and the transformation files can be very up to date, which means that if you do a very good calculation and then you can update it on a daily basis, and also you can extract that information very easily from the navigator files. So you will have a cryo-EM navigators, and you can also, during this process, import external coordinates points that you made on any other software. It doesn't have to be inside of the correlator. And then you just need the pixel coordinates point without any like units. And then we import it into Correlator, and then the Correlator will extract the stage transformation informations for you and will perform these transformations and then apply the stage correlation onto it. And then the final exported file, which is a navigator 2, can be directly used in the cryo-EM to collect data. So it actually provides a very flexible on-the-fly ways for the correlation, but it also supports the post-acquisition, which I use very often. And then also it's cross-platform, which means that as long as you have a piece coordinates file, you have Flint images, and it has a TIFF, MRC files or PNG, you can use it. And it's very user-friendly, as you can see. So the general process is that you will have your bright field, you have your Flint images and the navigator file, you put it into Correlator, do alignment transformation. So Correlator will also give you some assessment along the way, so help you to know whether or not your transformation is good. And you export the cryo-EM navigator file. And then there is an optional steps, which I generally use, is that I like to swap images, but you do need to consider the chromatic the aberrations between your bright field and your fluorescent channel, and also between different fluorescent channels. But in my case, is that generally it's very small. And then also I'm just trying to locate where I'm looking at in a larger area. And then after that, you can import it into the Cryo-EM. And then, so this is the actual demonstration on the fly, which is that we have your TN images and the FLIN images, and then we correlate. As you can see, the GUI on the left is that very user-friendly, and you get the navigator file imported. And one thing that I also sometimes use is that because the coordinates file you identified have the stage position already, so you can actually use it as your SPA coordinates where you collect data, which actually worked. And then, so this is just to go through all of the statistic and showing you that it's actually very close. So just some comparison with other software packages, because this field has been developed for years, and then everybody in the community contributes a lot in this process. And then I do find Correlator is very easy to use. And then also it does provide assessment and user-friendly, and more important is like on the fly. So it's ready for TEM. And then, like I mentioned, the GUI is a kind of important part of the Correlator, because when I first started, I do everything in scripting in MATLAB, and it ended up like very hard to actually translate that experience and those like errors and trials in the lab. So I collaborated with Matt, and then so we developed this GUI and the Correlator. So you can see that the whole function is very easy to use. And then also during this process, Matt also developed automated home central fighting process, and you can play with it, and also very easy to use. So we're talking about 3D correlation, which is everybody interested in doing. So 3D correlation is the same. In general, it starts with, instead of the source image being 2D, is source image being 3D. So you will have registration 3D and 3D, and you will get your transformation and applied onto your 3D, and then get the 3D transformation back into your 3D targeted image stack. And then, but for the FIB-milling, what happened is that you will have your source image, which is 3D very often, and then, but your SEN and FIB is actually one slice. One slice is a 2D image. It looks like 3D, but it's not as 3D. So people generally call it pseudo 3D. So this is a process where you have to do a 3D correlation to a 2D correlation. And then, so in this field, people have been working a lot to actually make this process better and better. So it starts with approach 1. So approach 1 is that you have your TN, you have your SEN image, you have your FLIN image. So using just one Z slice instead of the 3D Z slice, and you correlate that FLIN image to your SEN. And as you may remember from previously the transformation function, that the 2D affine actually present the rotation and translation in 3D, because especially in the projective transformation, where when you stretched along Y, it looks really like a 3D image that you projected onto it. And that's very much like your FIB image. So you will correlate that to get first the FLIN to SEM, and you will correlate FLIN, the correlated FLIN SEM to your FIB image. And your FIB image is where you're actually going to position your milling box. And in this case, you will actually have your Y, and Y actually present your thickness as where your Z positioning would be. And you will quickly realize that this applies very well for the very thin flat sample. It needs to be flat. And then also the marker and the fiducial on the same image plane, ideally will actually minimize the errors dramatically. So then based on this approach, the group actually move forward with what I call it approach 1+. It's also applied to the external cryo-EM system, where you would directly apply the FLIN image correlate with your FIB and SEM. So in this way, they are actually still using the coordinates-based transformation, but instead of using the 2D affine rotation and the translation in 3D, it directly uses 3D rigid bodies to determine. And because it doesn't have the translation information, so that the translation information is a little bit shaky. So to compensate for it, you need a lot of the beads, because they were using the nonlinear transformation, which is a least square nonlinear transformation. And then, so it is still like the more beads are better, flatter sample are better, and then also the precise registration identification is very important. However, both methods are unable to compensate for the movement of the RI during milling. And then the second approach, which has become more and more popular, is actually integrated film. So it starts with the integrated film, which is a wide field, and then it starts with at a different stage position, and then now people are moving to the coincidence point between the film, FIB, and SEM, where you don't need to move the stage as you guys are doing the Aquilose practical sessions. You will see that in order to collect the film images, you need to move the stage. But this recent development actually have the coincidence point, 3 channels, 3 modalities under the same spot. And then there is the confocal, because people want to get better and better resolution. And then they also wanted to actually directly transfer into the TEM. So make this process as smooth as possible. So it is very fast confirmation, and then but it does require hardware upgrade and configuration, and it's more suitable for integrated film data sets. So for us, we actually, because when we started, we don't have Aquilose 2, so we have Aquilose 1. And then we still want to do correlation. So how we're doing is that we need to combine both approach 1 and approach 2. Approach 1 offer you the initial transformation. Approach 2 offer you the on-the-fly adjustment. So we were thinking, how can we also get on-the-fly adjustment? So we were using the beads. So we're using a tiny little beads, a nanoparticle, to feed the cell. So the cell will take all of the beads. And because all of the beads will actually have relative positions next to your ROI, and those positions will change as your ROI is changing. So you can actually get the relative position while you are milling. And then also those positions will show up, the fiducia will show up on the SEM image. So that's actually help you to adjust on the fly. And then finally, you will take it out and then do TEM. So this is actually offer you a live monitoring during this process when you don't have an IFLM. And then so we also apply Correlator with the 3D CT box, which is I used also. And then so you can actually see that first we wanted to know whether or not the beads actually are good for the cells, because I do not want to kill my cells. And then after 4 hours at a reasonable concentration, is actually cells are very healthy. And then I wanted to know like what is the Z resolution it could possibly get. This is a wide field, because generally people using wide field to get actually faster screening and more data sets. And then I apply this approach. As you can see that under 200 nanometer thickness, you can still identify the beads. And then I have a better sets later to show. And then you can also see the beads under your TEM as well. And then, so I wanted to validate because later we actually got Aquilose with the IFLM inside. So what I was doing is that I'm still doing the same way. I wasn't using the correlation people are using right now. So I was still using the correlation based on the beads movement, but I kind of like using the IFLM as a confirmation to know that whether or not this method is working. So I don't do any adjustment based on the IFLM. I do all of the adjustment based on my SEM image and where the beads are relative to my sample. So what I found out that it actually works very well. And you can see the bead signals under SEM image all the way down to the thickness of the 250 nanometers. And then we take it out and then collect TEM. And we got the targets that we wanted, which is a cluster all the way of those mitochondria. And then we also applied this same methods combined with what in our lab developed called the Montage parallel array cryo-ET. So we are doing the montaging because as people mentioned, that limited field of view is really kind of like make the imaging harder and harder. So we wanted to know how we can actually capture both larger field of view as well as preserve the structural information, because you do not want to lower your magnification. And then so by doing this way, I first target where I want to target, and then I got the lamella, and then I applied a very classic impact, which is 3x3 tile sets, and then get this huge lamella, which is stitched together. And then the scale bar is 1 micron. So you can see that 1 tomograms at the resolution, which is the pixel size is 4.6, and you can have a really decent results. And then this is just a tomogram that you can see that going through all of the details shows up, and then you can see the lipid droplets. And then so here is just a little bit more background into the impact. So it's a regular Montage tile till series for the larger field of view at higher magnification. We do try different magnification even higher than that. There are some considerations, but it's applicable. So you just need to make sure your rotation, which is a translational shift, is reasonable, because otherwise it will your single tiles may not work. And then because we are applying a very moderate translational shifts for the dose distribution, so you can actually use all of those individual tile sets as we show in the D here, that you can get a stitch 3x3 Montage till series and then do segmentation and visualize the whole neighbors and then the context information. But you can also do the sub-volume averaging, and then based on the subtile till series, that you can get a reasonable resolution, which is in this case 20 angstrom. And we're not applying on the ribosome, we're applying on a kind of a more flexible targets. And then we also combine with tomographer. So tomographer, I will introduce a little bit more just in a second, is actually a simulation tool that you can help you to identify the best strategies to actually place a tile set, and then also to know the spiral translation. And then this whole process right now is very integrated with the Cryo-EM 4.1, where you can directly use a Montage multi-shot to actually get this going. And then, so this is a simulation. So I collaborated with Matt again. So we did this tomographers. What we wanted to do is that we want to simulate the till series collections process. So we can know that how much dose are actually accumulated at each areas, and whether or not this specific patterns will work to actually distribute the dose, and whether or not my ROI will stay in the center. So it actually works pretty well. The video just show you, like I quickly click all of the button. You can change it and to a different size and pixel size, and then the dose. So this is also a video while it's playing. It's just give you a better idea as what's happening. So you have a stitch Montage till series, and you can see the stitch Montage, the series is actually spiraling, and you can also have the tile. So the individual tiles are also spiraling, which is because we're distributing the dose, but the majority of the targets of interest stay where you wanted it to be stay, and then in the end you will get a reconstruction, just a regular till series, and you can pick the particle and then do the whole process. And then this is what we did, because we need to prove the proof of the concepts as to whether or not it's working. So we did it on the sub-volume averaging. We get a reasonably decent resolution, and then compared to the same pixel size that we collected using the regular ways, and it's very comparable. And then we also did more analysis as to, because right now you have a dose simulations, dose profile, so you can actually pick the particles based on your dose distribution, and then that can actually lead to a better resolution. And then moving from that, we wanted to actually combine the approach 1 and 2 together, but in a more efficient way, because not everybody wanted to feed the cells with the beads. And then right now, since we have access to Aquilose 2, which is with IFLM, so we collaborated with Thermo Fisher Scientific, Veronica, and then Tillman and John and Alex and Savada. And then we doing this process to actually optimize the correlation. So in this process, you will correlate IFLM with SEM, and then you will get your XY positioning. And then you will use that information, the Z-stack information from your IFLM 3D stack, to actually fine-tune your Z positioning in Y. It's actually a lot of more details, but you can see from the grammatic scheme, like everything is actually plays very well using the geometric transformation. And then finally, you will positioning on your FIB where to FIB at. And then so by doing this way, we are able to just pinpoint where we wanted to collect. So this is a very normal example that we used. And then so we were targeting the lipid droplets because they're round and very clear, so can actually tell us whether or not it's working. And then so we targeted and we move using IFLM at each stage to actually just trying to confirm whether or not ROI is there. If it's not there, and then we would not change it, because that's, you know, define the process. And then we move through this way, and in the end, we can actually still have our interest of the targets preserved in the whole process. And then this whole process is fully integrated in Autoton, as you see yesterday, because you will need to do the registration and then pick the targets and then go through the whole process. And then we also play with external cryofilm with aquilose. As I mentioned, external film is very powerful because you can actually collect it before the FIB-milling, and you can also collect it under confocal stack. So for this case, I just collected in our cryo-CLIM system, the Leica system, and I get the film stack. And then using the relative positions between the ROI and the fiducial, as I just mentioned, this position works very well, but you do need to make sure that the relative position is close enough because the lamella is moving. And then, so I only correlated once at the very first beginning, and using this geometric transformation methods that I mentioned above, the approach 4, and then I go all the way down in the auto 10, no manual intervention at all, go all the way down to 200 nanometers. I probably should intervene a little bit because it ends up to be very thin. And end up, because it's so thin, when during the loading, like transferring is actually the lamella break. So you can see that the lamella is break, but the fluorescent channels images are there. However, this method does have a limitation because your fiducial needs to be very close by your ROI, because the deformation of your sample during the handling and during the imagings cannot be counted for by your external film, which you collected before. So this is the acknowledgement. And then all of this work cannot be done without the support from the RILE Lab, and then also the Midwest Center and our collaborators. And then we do encourage people to download our tools and play with it, and then also check our Beanie Babies. Okay.
Thank you, Jay. Do we have questions? So I guess I'll ask one. So how long does it take to go through the process of developing the correlation tool to do your pre-correlation and then do the FIB-milling with IFLM and then go into the microscope to collect tomography data? You didn't comment on how much the metrics for collecting like a 3x3 array or things like that.
Oh, okay. I think I probably, yeah. So for the Montage, this one, it collected really very efficiently, because I think people probably know that there's another way to collect Montage Cryo-ET, which is using the hexagonal pattern. It's a very elegant way to do it because it really minimizes your dose, pre-dose irradiation damage, but it ended up having a really restricted beam size. And because of the beam size is so tiny, and then you have to pack them in a hexagonal way. And then because you pack it in a hexagonal way and plus your translation and rotation, you ended up only having one stitched Montage tomogram, which is gigantic, which in our case is also large when you stitch them together. But this way, you can actually collect them 1 hour. Generally, I think is like 1 hour to 1 hour and 30 minutes. You can actually have 10 till series and one stitched one out of that nice 3x3. And then the whole process is actually very automated because the stitching is based on the coordinates and it's a regular array. So it actually enables all of the pre-processings in a very streamlined fashion. So I would say like this part is pretty fast, but for the correlation, it takes time.
Hi, Jay. Lovely talk. So for this one, right, when you do subtomography on this Montage tomogram, do you extract subtomograms from the tile tomograms, or do you extract subtomograms from the whole big Montage tomograms?
So actually for this application, it's from the tile till series, because we were trying to see, like, first we see the ROI is preserved in the tile tomograms. And then so we wanted to just use a regular tile tomograms, and it's more efficient because the big one. So one thing about another methods, and we also encounter, is that the fully stitched one is gigantic. So it takes forever to run any process, but even extract the particles. And then, but if you use a regular one, it is the same time.
Can I ask a follow-up question? In that case, after you have the subtomograms, aligned in this tiled tomograms, right? Mm-hmm. Can you put them back in the big, montage tomograms?
It's actually a very good question. You should be able to, because you will have the coordinates file, and then you will have the coordinates shifts. I haven't tried it, but it's a very good question. Yeah, that would be nice if you do it and then apply back into the big montage. But there are some, shifts to apply during the correlation while you're tile stitching. So, yeah.
It will be beautiful if you have it.
Yeah, that would be great.
There's a couple of online questions. I'll start with the first one, which is come to the IFLM usage. Is the fluorescence imaging done automatically, kind of in an atlas-like procedure, or as a user, do you have to navigate and collect images at all positions? And how does focusing work with IFLM?
Oh, for IFLM, so I think people will quickly notice that is right now is quite manual, unless you wrote the scripts, which you can, and actually to collect montages. But the IFLM, especially for the Thermo Fisher Aquilus 2, you will notice that it has a pretty large field of view, and you can see I would say 4 squares per field of view, so that definitely helped. And there's some ways to move the stage right now, but yes, it is manual.
And then there's another question about, do you use any pre-processing prior to doing the registration, such as do you go to use tools like the LaserX tool?
Yeah, so it depends on how noisy your image is. I guess is that generally people would want to know how good the registration points needs to be to achieve the better correlation. If that's a question, I would say the more precise, the better. But it is to a point is that I think it's more important. You need to make sure it is corresponding, matching points, so that you don't misplace the points where on the IFLM and then the points on the TEM.
A couple more online questions. There's one, can impact be combined with bisect?
I never tried it. I think is one of the optimization that we're trying to do right now is actually to better, because we're using the tracking as well. So I think there's an image to actually show the whole process is the tracking is actually also the focus area. So right now there's one thing that we would like to optimize, even though it still works for a while, is that we can actually optimize the tracking for each tile set by using that. And then that is one of the things right now people are applying with the pace and then the bisectors that you use in one area, doing the geometric projections and then help with the tracking. So I never tried it, but that should be optimized, yeah.
And then I guess one last question from online. How well can you resolve organelles bound to protein supercomplexes through lift out, or is the waffle method preferred? I guess this is going back to just general FIB-milling.
So, sorry, what is your question?
How well can you resolve organelles bound to protein supercomplexes when using lift out, or is the waffle method preferred?
I think is a question more for the lift out. I don't know. So I would say for the complex, I guess from the standpoint of the correlation, and then it really depends on how small and how big your target is. And then that can help you to identify which way is better. And then also the thickness of your lamella. And I know that people keep telling you that, oh, the thinner is the better. But there's more studies to show you that you cannot go beyond certain thickness of the lamella. It's going to damage your structures. And also you're going to lose the volume information that you wanted to preserve in your TEM. So it's not always the thinner the better, unless you are looking just at like a tiny little protein.
So I have a question, like Veronica gave the practical, be careful doing this, wear a mask all the time, no breathing. Do you have recommendations for how people should work when they're doing correlation workflows?
It generally is the same, but less, more breathing for sure, because I did lift out, and it is like the contamination is a huge thing. But for this way, just general cryo handling that you clean nitrogen, like dry tools and everything. And then also the one thing that I think is very important is the system optimization sometimes comes to play that people often forget, is that your cryo stage may have some, optimization that needs to be done. And then that needs to be done in a very rigorous way, which means that you assess and then you actually load to the TEM and go back and then talk to experts and figure out what is going on, so that in a way you can actually do a 2D correlation. Because right now a lot of people are kind of scared to do the 2D correlation because the 2D images, you never see people show 2D images because it has a lot of contamination.
Any other questions? Any other online ones?